See, it was the first hormone to be isolated from animal source, in a form that could be administered therapeutically to humans. First hormone isolated from animal source. For what purpose? For therapeutical purpose. And then the first hormone to have its primary and tertiary structure determined. Then the first hormone to have its mechanism of action elucidated. And the first hormone to be measured by radioimmunoassay. The first hormone known to be synthesized from a larger precursor, that is the prohormone. And finally, the first hormone to be synthesized with recombinant DNA technology. So you can see how many firsts are associated with insulin. First, hormone to be isolated from animal source, and having the primary and tertiary structure determined, then having this, it's a mechanism of action explained, and so on. Okay, now these are not important for the exam point of view, or say, just if you know, better to know. So the key thing which we will talk about insulin now, synthesis of insulin. So in the synthesis of insulin, let's say insulin is a peptide hormone. We have already said that, and a peptide hormone consisting of two straight chain. That is an A chain having 21 amino acid, and B chain having 30 amino acids. Two disulfide bridges, they link the A chain to B chain, linking the A chain to B chain, and third disulfide bridge is located within A chain. So synthesis of insulin, directed, and the synthesis is directed by a gene on the chromosome 11, which is a member of superfamily of genes that encode related growth factors. Chromosome 11. Then the mRNA directs the ribosomal synthesis of what? Prohormone, we said. So pre-pro insulin. The first hormone to be synthesized from the prohormone. So the pre-pro insulin, and this pre-pro insulin contains 4 peptides: a signal peptide, the A and B chains, and the connecting C peptide. So the signal peptide is clipped early in the biosynthetic process, while the peptide chains are still being assembled, which yield the proinsulin. And then the proinsulin is then shuttled to the endoplasmic reticulum, brought to the endoplasmic reticulum, where with the connecting peptide is still attached, the disulfide bridges form to yield the folded form of insulin. And then proinsulin is packaged in secretory granules on Golgi apparatus. So shuttle to endoplasmic reticulum. Now the proinsulin packaged in secretory granules on the Golgi apparatus, and during this packaging process, proteases clip the connecting peptide yielding insulin. So basically, when you say the synthesis of insulin, then what happened? From pre-proinsulin, proinsulin was yielded, and then from proinsulin, through these processes, taken to endoplasmic reticulum and packaged in secretory granules on Golgi apparatus. That is, this finally the insulin is yielded. So insulin and the clipped connecting peptide are packaged together in secretory granules, and when the beta cell is stimulated, they are released in equimolar quantities. Okay? So equimolar quantities into the blood. You can understand equimolar if you know the concept of mole and equimolar. So the secretion of connecting C peptide is the basis of test for beta cell in person with type 1 diabetes mellitus, who are receiving injections of exogenous insulin. And insulin is metabolized in liver and kidney by enzymes that break disulfide bonds. Tell me one thing, have you gone through this synthesis of insulin in biochemistry or any other subject?
No, sir.
No? Okay. But I assume that biochemistry will deal with the synthesis process in detail at least. So anyhow, so what are the key informations here? If you can list those, if you have to list the major events, which one we talk in this process of synthesis of insulin, what are those? Anyone can list those factors. What are those factors? Or say events, major events? Pre-pro, you have to remember that there is a pre-pro insulin. If you remember those things, the key points, then you can correlate those things. Pre-pro insulin, from those pre-pro insulin, pro insulin, and then the pro insulin in subtle to this endoplasmic reticulum, and then the secretory granules, the Golgi apparatus, and then the insulin is yielded, and then still this connecting C peptide is there, connecting peptide. So that is one of the basis for the test of the type 1 diabetes mellitus, and the person who is exogenously administered with this. Okay, now you can see the A chain and B chain are released, now inactive and excreted in urine. So when you see the regulation part of this insulin, then you can say the insulin secretion and the beta cells, glucose, GLUT2, glucose transporter 2, and then the glucose by this action of this glucokinase converted to this glucose 6-phosphate, and then oxidation is there. And now this event of this calcium influx is there, calcium increases, and finally the exocytosis is there. Okay. Now, when you talk about the factors affecting insulin secretion, then you can see there are a list of factors which stimulate, and then the list is short, but there are factors which inhibit this insulin secretion. So what are the stimulatory factors? Increased glucose concentration. Inhibitory factor, decreased blood glucose. Then increased amino acid concentration. Fasting is inhibitory factor. Stimulatory, increased fatty acid and ketose concentration. Glucagon, cortisol, glucose-dependent insulinotropic peptide, GIP, potassium, vagal stimulation, and then certain drugs. And obesity is also one of these stimulatory factors for insulin secretion. Whereas inhibitory factors, along with this decreased blood glucose, fasting, exercise, somatostatin, then alpha-adrenergic agonist, and digoxide. These are the inhibitory factors for insulin synthesis. Now, when you talk about the action of insulin, then the first thing that comes to our mind is that insulin is hormone of abundance. Why hormone of abundance? Let's see. When there is availability of nutrients, availability of nutrients exceeds the demand of body. Hey, what is happening? Availability is exceeding the demand. So this term abundance is when nutrients are available in abundant quantity. Insulin ensures that excess nutrients are stored as glycogen in the liver, tissue, and as protein in muscle. Now, these stored nutrients are then available during subsequent periods of fasting to maintain glucose delivery to brain, muscle, and other organs. So, whenever there is the supply or availability is more than the demand of the body for maintaining normal homeostasis of body, normal physiology of body, then that is stored. What is stored? Nutrients are stored as glycogen in liver, fat in the adipose tissue, and protein in muscle. And whenever there is requirement, then that will be provided or utilized, because supply to the brain and the glucose supply to brain, muscle, and other organs, and glucose being one of the immediate source of energy, that is very important. So insulin has following actions on liver, muscle, and adipose tissue. What are those? Decreases blood glucose concentration. The hypoglycemic action of insulin can be described in two ways. It causes a frank decrease in blood glucose concentration, and insulin limits the rise in blood glucose that occurs after ingestion of carbohydrates. So suppose a condition when insulin is not there. Okay, what will happen? Assume a condition in which insulin is not there, what will happen? You keep
hyperglycemia.
Pardon?
There will be increased glucose level in the blood.
Yes, definitely. Because we will keep on ingesting, and with the ingestion of food, the carbohydrate concentration, or say basically this the glucose will keep on increasing, but insulin is limiting that by this storage. So the hypoglycemic action of insulin is the result of coordinated responses that simultaneously stimulate glucose oxidation and inhibit gluconeogenesis in these manners, and as follows. Like you can see, insulin increases the one which we saw in the figure, and the insulin increases glucose transport into target cells such as muscle and adipose by directing insertion of GLUT4. There was GLUT2 for the revolution here for storage, and the GLUT4 into the cell membranes, and as glucose enter the cells, the blood glucose concentration decreases because now they are stored in cell. Insulin promotes the formation of glycogen from glucose in the liver and in muscle, and simultaneously inhibits glycogenolysis. That is the glycogen breakdown, and insulin inhibits gluconeogenesis, synthesis of glucose, by increasing the production of fructose 2,6 biphosphate. And what will that do? Increases phosphofructokinase activity. So in fact, the substrates are directed away from the formation of glucose. Now, second major action of insulin decreases blood fat and fatty acid and keto acid concentration. So the overall effect of insulin on fat metabolism is to inhibit the mobilization and oxidation of fatty acids and increasing the storage of fatty acid. So as a result of that, this blood fatty acid will decrease, because they are being stored, acting as reservoir these cells, and as a result, insulin decreases the circulating level of fatty acids and keto acids. Then in adipose tissue, insulin stimulates fat deposition and inhibits lipolysis. So that is one mechanism by which the decreasing and the blood fatty acid, and because they are stimulating fat deposition and inhibiting lipolysis. So yeah, you can say simultaneously insulin inhibits keto acid, that is beta hydroxybutyric acid and acetoacetic acid formation in liver. Why? Because decreased fatty acid degradation. Fatty acid degradation has decreased, means that less acetyl coenzyme A substrate will be available for formation of keto acids. So this is how insulin play a role in maintaining the blood keto acid concentration. How? Because it is making less acetyl coenzyme A available for formation of keto acid. Then the third important action of insulin is to decrease blood amino acid concentration. We have seen its action on the glucose, its action on fatty acids and keto acids. Now amino acid concentration. So insulin and the effect of insulin on protein metabolism is anabolic. What is anabolic? What is anabolism? Okay. So insulin increases amino acid and protein uptake by tissues. So here, one thing you must have noted that whatever we said, decreasing glucose, decreasing fatty acid and keto acid, and decreasing this amino acid concentration, we are not saying that insulin is removing them. No, it's not removing them. It is helping in storage of these things in tissues, so that the level in blood is decreased. So basically, it is decreasing their level in blood. Okay, so insulin stimulates amino acid uptake into target cells like muscles, increases protein synthesis, and inhibits protein degradation. So anabolic action and protein metabolism. Anabolic action, the increasing protein synthesis, inhibiting protein degradation. So major actions of insulin and the effects on blood levels. Here you can see, by increasing and this glucose uptake into the cells, that is decreasing blood glucose, increasing glycogen formation and decreasing the glycogenolysis, decreasing gluconeogenesis. All this will decrease the blood glucose, and then increases protein synthesis which will decrease blood amino acid, the amino acid level will decrease in blood, and increases fat deposition, decreases blood fatty acid concentration, decreases lipolysis, decreases blood ketosis, and then increases potassium uptake into cell, that will decrease blood potassium level. These are the major actions, or say major functions of insulin. Along with these functions, there are other functions also. Like you can see, in addition to the major actions on carbohydrate, fat, and protein metabolism. So you can imagine how important insulin is for metabolism, because it is playing role in metabolism of all these major macronutrients like carbohydrate, protein, and fat. And there is one saying that if you take care of carbohydrate, protein, and fat, other things will be taken care of. So, along with this, here you can see insulin promotes potassium uptake into cells, and at the same time promotes glucose uptake by increasing the activity of sodium potassium ATPase pump. And this action can be viewed as protecting against an increase in serum potassium concentration. So when potassium is ingested in the diet, insulin ensures that ingested potassium will be taken into the cells with glucose and other nutrients. So it has insulin appears to have a direct effect on hypothalamic satiety center. Hypothalamic satiety center, independent of the changes it produces in blood glucose concentration, by maintaining the level of potassium concentration and serum potassium concentration. Now, pathophysiology of insulin. If you have already gone through this pathophysiology of insulin, we will not talk about this in detail in physiology. Have you gone through this in pathophysiology of insulin?
No, sir.
Okay. Okay, then we will talk in brief about this. So, you must have some idea about what will happen. There is a problem with insulin. Diabetes, right? Okay. So major disorder involving insulin is diabetes mellitus. Okay. Not diabetes insipidus. Right? Is insipidus related with this?
No, sir.
Then?
Related with ADH secretion.
Okay, yes, good. So diabetes mellitus, in this diabetes mellitus, there is type 1 and type 2 diabetes mellitus, and then insulin-dependent IDDM, non-insulin-dependent diabetes mellitus, and IDDM. Okay, so in one form of diabetes mellitus, type 1, there is, what is happening? What is the difference between type 1 and type 2? In type 1, there is inadequate insulin secretion. There is problem with secretion. Whereas in another form, type 2, there is problem with insulin binding with its receptor and showing its action. So another form, type 2, there is insulin resistance of target tissues. So insulin-dependent diabetes mellitus or type 1 is caused by destruction of beta cells, and that's why there is problem with insulin secretion in this type. So often as a result of autoimmune process, that means destruction of beta cells. So when pancreatic beta cells do not secrete adequate amount of insulin, there are serious metabolic consequences. Like, and these consequences, all of these are related with the functions of insulin which we talked just now, like metabolism of carbohydrate, fat, protein, they will be disturbed. Then, type 1 diabetes mellitus is characterized by following changes. What are those? Increased blood glucose concentration. So, insulin is not there to maintain this level of blood glucose. So, there is increased blood glucose concentration from decreased uptake of glucose into cells, decreased glucose utilization, and then increased gluconeogenesis. And all of these are associated with the functions whatever we have talked just now, functions of insulin. Then increased blood fatty acid and keto acid concentration. I think I don't need to explain you all these, because these are all, whenever there is problem with the secretion, insulin level decreases, then those functions will be altered. As you can see, increased lipolysis, that insulin is decreasing lipolysis, so that is not there, or say in decreased concentration, then increased lipolysis, increased conversion of fatty acid to keto acid, and decreased utilization of keto acid by tissues. Then increased blood amino acid concentration from increased breakdown of proteins into amino acid, and there is also loss of lean body mass, that is catabolic state, and loss of adipose tissue, because we have seen that it was involved, and it is involved in anabolic action. Then second type, non-insulin dependent diabetes mellitus. I will not go into the features, and then there's the treatment process. No, I will talk only those things which are relevant for physiology, because I know you will be pathology will deal all those things in detail. So I'm just talking about from physiological point of view, and this alteration of functions. So non-insulin dependent diabetes mellitus or type 2 is often, that is often associated with obesity. Like it exhibits some, but not all, of the metabolic derangements seen in type 1 diabetes mellitus. Why not all? Anyone would like to answer? Why not all? We said it exhibits some of those metabolic derangements seen in type 1 diabetes mellitus, but not all of them. Why?
Because there is relative deficiency of insulin, not complete deficiency.
Yes. And in the previous case, there was problem with secretion. In this case, there is problem with the resistance on the target tissues. So insulin is available. So whichever some of those tissues working, and then they are showing their action, receptors are available and they can bind. So type 2 diabetes mellitus is caused by down regulation of insulin receptor in target tissues and insulin resistance. What is down regulation of receptors?
Decrease in number and sensitivity of insulin receptor.
Decrease in number. Okay. So basically that is decrease in number. Number is more than sensitivity. There is when you say down regulation, then decrease in number is there. Can receptors be upregulated? Yes or no? Here, there is down regulation of receptors. So my question is there conditions in which receptors are upregulated, their number increases?
Yes, sir. Do it.
Yes, there are conditions when receptors are upregulated, their number increases. And there's more receptor, and that happens usually when the concentration is low, so utilize most of them, Okay. Anyhow, so basically what is happening? There is downregulation of receptors, and the target tissue, there is insulin resistance. So insulin is secreted normally by beta cells in this case, but at normal concentration, it cannot activate its receptor on muscle, liver, and adipose tissue. Thus, insulin is unable to produce its usual metabolic effects. So whatever we have talked till now about many hormones, whichever hormones we have talked about, we have seen that to show its action, these hormones, they need to bind to the receptor, and then this hormone receptor complex is formed, which initiates this second messenger system, and then their actions are shown. So there is problem with receptor, so their functions will not be shown. So typically blood glucose concentration is elevated both in fasting and postprandial, that is after eating states. So here I got this figure from internet, and I found this simple one to explain you what happens in these cases. Here you can see this decreased glucose uptake and utilization is will cause this hyperglycemia in urine, glycosuria, and this glycogenolysis, then protein catabolism and gluconeogenesis. So there is glycosuria and osmotic diuresis, and as a result of that, there is polyuria, then polydipsia, and polyphasia. So 3P, polyuria, polydipsia, and polyphasia are there. And then problem with this lipolysis and ketogenesis. So lipidemia and in blood, lipidemia and ketoacidosis is there. In urine, ketonuria, and loss of sodium, potassium, and electrolyte imbalances, acid-base imbalances are there. Signs and symptoms, there is acetone breath, or say sometimes this fruity smell, and then hyperpnea, nausea, vomiting. So these are just the signs and symptoms, the features. I will not go into the details of these. Okay. So physiologically, this is more important. What happens and how its level is maintained. Like you can see, when the plasma glucose concentration is increasing. Plasma glucose concentration increasing, then this on beta cells, and beta cells stimulated, and beta cells of pancreas. So when beta cells pancreas gets stimulated, then there is increased insulin, and this insulin will show its action on liver, so its action on muscle. So on liver, it is increasing glycolysis, increasing glycogenesis, and increasing lipogenesis, whereas on muscle and other adipose cells, it is increasing glucose transport, and by this mechanism, this will decrease the plasma glucose concentration. And when this plasma glucose concentration level is maintained, then now there is feedback to beta cells, and now the beta cells will know that this concentration is now maintained, so no more, also no further increase, and it will keep on secreting insulin in normal rate, but increased secretion synthesis is stopped there. Okay, so this is how the level is maintained. So this is just synopsis, showing this synopsis of insulin, in which you can see the cells of origin beta cells, structure 51 amino acid peptide, biosynthesis and from the typical peptide, transport in circulation dissolved in plasma, half-life 5 minutes, factors affecting release, mainly the plasma glucose level. Then the GI hormone to this feed-forward reflex, and this parasympathetic amplification. Parasympathetic effect will amplify, sympathetic will inhibit, and then target cells, liver, muscle, adipose tissue, and this target receptor, membrane receptor with tyrosine kinase activity. Then whole body or tissue action, decrease plasma glucose by increasing transport into cells and increasing metabolic use of glucose, and action at cellular level is increased glycogen synthesis and aerobic metabolism of glucose and increased protein and triglyceride synthesis. Okay, so I think as a start गर्ने नगरौँ है, अब यो टाइम पनि भयो, अब फेरि बीचमै गर्नुपर्छ हैन. Okay, for next class I have an assignment for you. Hey, के भनेर सोध्नुस् न. Hello.
Yes, sir.
Yes, sir.
Assignment के भनेदेखि तपाईहरु सबैजनाले insulin glucagon molar ratio, why it is important, why it is clinically important, just look up to that and just read it. I don't need any, you don't need to submit any write-up or anything like that. You just read it so that you will understand it better when we discuss about this. Okay. हो भन्नुस् न, यति सजिलो असाइनमेन्ट दिए त मैले.
हुन्छ सर.
ओके ल, थ्यांक यू.
एउटामा यस्तोमा अस्ति बाँकी थियो कि आउँदै आउँदो अरे बल्ल आज आयो क्या